Weeks 7 and 8 are going to talk about the whole subject of ecography. It's a terrible name, but it encompasses a whole range of rules that express ecological concepts over huge geographic scales or huge temporal scales. The relevant reading is right there, shown to you, the parts of chapter 15. I was going to include a separate reference for you, but I've decided to back off on that. Look at this diagram over here. You see these are primate families, so apes and monkeys, and it is graphing geographic range versus density. So, and look, that geographic range is in square kilometers and it's a log, so these can be huge. And see, it's something that is done then by not just one person, but typically a person then mining out the literature. This began as a study that was called aerography. We don't really use that term too much anymore, but this looked at like these salamanders that are shown here, various sizes, shapes, other features, and their geographic ranges. Experiments were not possible, but it was just comparative work. This eventually leads to what we call it today, which is macroecology. So if you see an ecological work that has something like this, a map of the globe and a huge geographic range, it's probably falling under the notion of macroecology. General patterns from large scale data. And part of this, the trouble with doing something like this is that the scale of the ranges can be tremendous. So that cute little guy up there is the devil's pupfish, and it just lives in a couple of different springs in one part of the world, and that's its total range. Compare that to the blue whale, which is a large migrant in the world's oceans that has a range of hundreds of millions of square kilometers. Well, all of this type stuff influenced years ago this fellow named Eduardo Rappaport. So he wrote the book called Aerography, and he was one of the first to do this type of stuff. He compared the size of range to just about everything else, typo, just about everything else that he could. Diversity, latitude, body size, etc. And he made a whole bunch of things wherein range size was graphed against these different factors. One of them had also been worked on for many years by this fellow named Willis, and he called these things hollow curves. And Eduardo Rappaport was fascinated with hollow curves. So it starts there. And look at these things. They typically show some vertical scale of the number of taxa, in this case number of species collected, and then some type of measure of geography or a proxy. And one of the things that you see, if you look at this, so look at this one here, number of individuals collected, number of species collected. So you have lots of places or samples where only one individual is collected of the many species that are shown there. Here's a better way to look at it. Consider it. These things are almost always unimodal. They are right skewed, and this says that most species tend to have very small geographic ranges. So if we look through these things, this is actual data based on real data, but I don't have it scaled here for you or listed for you. But notice, area of geographic range increases towards the right, and the proportion of species or percentage is over here. There's your hollow curve. Here's another example. Number of species, geographic range size. So many species have small geographic range sizes, and that decreases as you go to organisms that have huge geographic range sizes. So lots and lots of pupfish type creatures, very few blue whale type creatures. Here's another one. Sort of this is number of species and the number of rivers that it's found in. Again, many species are found in one river, and successively less and less are found in many rivers. So see, that would be a proxy for geographic range size. And then just another one showing the same thing here, though it's more GIS oriented as far as number of occupied grid cells. There are, or there have been found, lots of different variations in hollow curves. Birds have certain features, reptiles have different features, mammals are all over the thing. And then there's even variation by continent. So the average, so here's range size in Australia versus range size in North America, and just sort of compare range size of turtles in North America, about 0.6, range size in Australia significantly less than that, just a little bit over 0.2. Bats, range size in North America, almost 1 in Australia over here. This led him to come up with what's now called Rappaport's rule. And simply stated, in general, geographic range size gets larger as latitude increases. So the closer you go to the poles, the more species you have that have huge range sizes. Now notice, this is an interesting trend that kind of goes, it follows latitude and the latitudinal diversity gradient, which is sort of shown here. So the light gray line is species richness. Notice it is declining as you go towards higher and higher latitudes. That's Humboldt's latitudinal diversity gradient. But the geographic range increases at the same time. So less and less species having bigger and bigger ranges. And you get this sort of scissoring type diagram. And then here's just another one. Again, in your book, these are based on actual species data, so you can see those things. Rappaport's rule has been extended to other places now. So it's extended to elevation, so that the higher one gets in elevation on a mountainside, the less species there are. Remember, we've talked about the latitudinal diversity gradient extended that way, but the larger those fewer species ranges are. Same thing with marine depths. As you go deeper into the ocean, there are less species, but they have larger ranges. And so this is just a picture of the elevation equivalent of Rappaport's rule. As you go higher and higher on a mountainside, the light gray line shows less and less richness. That's the elevation equivalent of the latitudinal diversity gradient. And then here's this scissor type diagram showing the geographic range. So fewer species have wider ranges. Many hypotheses or ways to think about this, and I'll leave this to you as far as reading this in the book. Now let's finally get to ecography itself, which, as it says there, geographic variation in ecological characteristics, processes, what have you, has led to the development of all of these rules. Not laws, rules. So first of all, here's the one that usually starts our study of this. Body size, mass, increases with latitude. So animals get bigger the farther north or south one gets away from the equator. And this diagram here even shows this works with humans. So African humans, which have they're predominantly present in Africa at lower latitudes, which means higher temperatures, have lower body masses. Compare that to Europeans, where they are present at higher masses at these lower temperatures. And then this diagram just sort of shows two deer, Nicaragua, Michigan, again, tropical for Nicaragua, temperate, high temperate for Michigan, a larger body mass. Moose in Sweden. Sweden covers it's a huge range of latitude from 50 degrees north latitude to 70, and notice this adult body mass, it increases with latitude, and this is shown by both males and females. The why for Bergman's rule, it typically is said to be an issue of maximizing the volume while minimizing the surface area. Doing that means you retain more heat. And so just the standard box example comparing volume and surface area. Basically, getting big is good in colder temperatures where there is a heat budget deficit for the Earth so that the animal living there can retain more heat. There are other possible hypotheses. The book talks about several, so I'm going to direct you to read the book, and I'm going to give you the helpful dude tip here that you should know, meaning understand, meaning be able to explain at least one of these for exam 2. Bergman's rule, you can look at it from a paleontological perspective as well, or a time perspective, and think about this. For the last 18,000 years, we've been sort of through a glacial interglacial cycle. We had a maximum extent of the glaciers about 18,000 years ago, meaning no animals would be living on top of this mountain of ice that covered the northern hemisphere, and then that slowly receded, and these animals had to recolonize. So we are sampling animals that have recolonized over this short, less than 20,000 year period, and yet they obey this Bergman's rule, meaning this Bergman's rule gets set up very quickly. And then just a couple of diagrams. I'm not going to sort of worry about these so much, just to show you things. Let's go to this Drosophila, Drosophila subobscura. It's the fruit fly. This has only been introduced in North America for a couple of decades, and so you see European males and European females, and their following of Bergman's rule. And then look at North America. Again, it's only been here several decades. Now granted, Drosophila have really rapid generational cycles, so evolutionary changes can be made quickly. Notice it obeys Bergman's rule as well. Here's a different one, Allen's rule. This is kind of an oddball thing. It refers mostly to mammals or exclusively to mammals, and this says that appendage size, any part of the body that extends away from the torso, increases as temperature increases, which means then that there would be a rough correlation with latitude. The lower picture is the classic bunny that we see around here eating our garden, but the upper picture is the jackrabbit. That's more common in places like Texas and Arizona, and notice the ears are longer. The front and hind limbs are longer. Even the tail, you can't really see it on this cottontail here, but even the tails are longer. Just some other pictures, so sort of rabbits and foxy things in low or high latitudes, and then as you get to lower and lower latitudes, bigger ears, mostly what's shown. Glosier's rule, this is kind of an odd one as well. This says that if you have closely related species, so maybe species in the same family. So here we have two members of the family Ursidae, the bears, includes all bears. And so you see a grizzly and a polar bear, and as you look at humidity increasing, you have a darker color. So humidities are notoriously low in polar areas where that polar bear exists, and they're much higher where grizzlies exist, and so you see this color variation. It's kind of a minor rule. Here's an interesting one from the oceans, Jordan's rule. As you go up in latitude, again, so facing or heading towards the poles, the size of the fish increase. That's part of Bergman's rule, but even for some groups where there is no size increase, there is an increase in the number of vertebrae. So here is this genus Galaxius, species Brevipinus, and notice as latitude increases, there is an increase in the mean number of vertebrae. This is the same species with different numbers of vertebrae depending upon latitude. Let's sort of look lower in the developmental area. Thorson's rule. Thorson's rule looked at the ratio of planktotrophic versus leucothotrophic larvae versus latitude. So first of all, leucothotrophic larvae, these are what are sometimes called direct development. These organisms, as they hatch from the egg condition, they are endowed with a large yolk sac. So they have food. They have lunch packed for them for potentially weeks, maybe even over a month or so. So they don't swim or float a long time before they settle. So there's a restricted dispersal for these things. This is contrasted with planktotrophic larvae, pelagic development, which are given no yolk sac or a very small one, meaning they must feed in the plankton. So they spend much longer times actually living up in the water column, eating and growing larger there before they disperse. If you look at this, pelagic development, planktotrophy, direct development, leucothotrophy, notice one decreases with increasing latitude, one increases with increasing latitude. You can think about the arms race and geography. So within the same species, within the same related groups of families or genera, etc., or just overall in general, just counting things and looking at them, if you look at the tropics, organisms typically have all kinds of anti-predatory defenses. Spines, thickened shells, bright camouflage colorations, things like that, that decreases as you go north in latitude. So the pictures here are showing a highly spinose, ornamented snail and clam, and they're from the tropics, clearly, because they are covered with these spines, these anti-predatory devices. By the time you reach the polar areas, near the Arctic and the Antarctic, those same type things, clams and snails, are pretty boring and dull. Smooth shells, not overly thickened in any particular way, no sharp concentric ornament or radial ornament. The island rule, sometimes this is called Foster's rule, and this one's kind of an odd one, but it says that if you've got organisms that are large on land, if they somehow get isolated on or colonized to a much smaller area, an island, smaller microcontinent or whatever, then to adapt to this, they will shrink in size over time. So, for instance, the pygmy mammoths of the South California Channel Islands, even among ancient humans, the Homo floresiensis, these so-called hobbit humans found on an ancient island environment this way. Sometimes it goes in reverse. If you have species that are originally small on a large continental mass, if they get isolated on an island, then they become much larger. And the island rule now has been extended into the deep marine. So if you've got organisms that are large in a shallow area, very often they are small in the deep, or the reverse. And so here's some snail examples, and then these large isopod here. I put the paleo question mark there because people are trying to show that this also occurs for paleo, and indeed it does, and you'll probably see, wink wink, something like this on exam 2.